Welcome, class, to Chapter 6, where we'll talk about the extracellular environment. The extracellular environment includes everything that's located outside of cells. The cells will receive nourishment from the extracellular environment and release wastes into the extracellular environment. And cells will also communicate with each other by secreting different chemical regulators into this extracellular environment. About 67% of our water is within cells, and 33% is in the extracellular compartment outside of cells. 20% is in the blood plasma, and 80% makes up what is called tissue fluid or interstitial fluid. So here's a look at the extracellular environment. Again, anything outside of cells. So we'll have our collagen, protein fibers, ground substance. We'll have our interstitial fluid. The extracellular matrix contains protein fibers of collagen, elastin, and a gel-like ground substance. You can read here what they each do. Protein fibers provide support. The ground substance is made of some glycoproteins, and integrins and glycoproteins that extend from the cell cytoskeleton bind to the extracellular matrix. So what are integrins? They will impact the polarity of cells. They affect adhesion and motility of cells, and they also affect how cells proliferate or reproduce and make more of. The plasma membrane permeability, we'll get into transport now of moving things into and out of the cell. The plasma membrane is what we say selectively permeable, meaning that it allows some molecules to cross but not others. Generally, it's not permeable to proteins, nucleic acids, or anything too large to get through, but it will be permeable to ions, nutrients, and wastes. There's categories of membrane transport. We say there are non-carrier-mediated ways and also carrier-mediated. The non-carrier-mediated are simple diffusion of lipid-soluble molecules, simple diffusion of ions, and simple diffusion of water, which is osmosis. Carrier-mediated means there's some sort of carrier protein helping something get into or out of the cell, and these are facilitated diffusion and active transport. Energy involvement can either be passive or active transport. Passive transport are molecules moving from higher to lower concentration without using metabolic energy. And active transport are when molecules move from lower to higher concentrations using an ATP and specific carrier pumps. So these are different types of transport, and you can see them listed here. These are non-polar molecules, are able to simply diffuse through the membrane. Inorganic ions and other large substances might need the help of channel or carrier proteins to get them through. And here are small organic molecules using a carrier protein to get through the membrane. Diffusion and osmosis. So some of this will hopefully be a review of what we went over in lab. A solution consists of a solvent, which is most likely water, and a solute, which are molecules dissolved in water. The molecules in a solution are in constant motion, and if there's a concentration difference between the two regions, random motion will reestablish equilibrium by diffusion. So things will move from an area of higher concentration to lower concentration, and this is what the second law of thermodynamics says. So this is just a look at diffusion of a solute. It'll always move from an area of higher to lower concentration until there's equal concentrations of the solute on both sides of the membrane. So diffusion will occur without any physical separation or across a permeable membrane. Net diffusion is due to random movement, and the net direction of diffusion is always from high to low solute concentration. And the mean diffusion time is the average time it takes for a solute to diffuse. So this is just an example of proteins, small diffusible molecules and ions and glucose, and you can see them listed there. And it just shows you the direction that these molecules will travel. You can see that the proteins are too large to get through the membrane of this balloon. The small diffusible molecules and ions will leave the balloon because they'll go into an area of lower concentration. And the glucose that's in the beaker, it looks like it has about an equal concentration within the beaker solution and within the balloon, so it's going in both directions. Diffusion through the plasma membrane. This talks about things that are able to get through the membrane and things that are not able to get through the membrane. Gas exchange is the net diffusion of oxygen into cells and carbon dioxide out of cells due to concentration gradients. And this is extremely important because all of our cells need oxygen and they will create carbon dioxide as a waste product through cellular metabolism. And water will pass through using special channels called aquaporins in a process that you guys should hopefully be familiar with called osmosis. So again, this is a look at oxygen moving into the cell through diffusion because the inside of the cell has less oxygen, and carbon dioxide will exit the cell because carbon dioxide will go into an area of less concentration. This will talk about how charged ions can pass through using ion channels, and larger polar molecules can usually not pass through at all, but they need special carrier proteins for the help of them because they're larger. And this is a look just at a channel helping ions to get through the cell membrane, and it will be open or closed. The rate of the diffusion is measured by the number of diffusing particles, and we did cover this in lab. It depends on the magnitude of the concentration difference, the permeability of the membrane, so how well things can get through, the temperature of the solution. A higher temperature increases the rate, and the surface area of the membrane. If there's increased surface area, usually due to little processes called microvilli, diffusion will also increase. Osmosis is simply the diffusion of water. Water can pass through the plasma membrane slowly because it does not have a charge to it. And we give it a special name called osmosis. Usually it's aided by channels called aquaporins. Osmosis, there must always be a solute concentration difference on either side of the membrane, and the membrane must be impermeable to the solute, meaning the solute must not be able to get through the membrane, only water can get through. So here's a model look at osmosis showing the semi-permeable membrane and showing that the solute, let's say it's glucose or salt, is too big to get through the pores in the membrane, but water is able to get through. And water will always travel into an area where there's more solute particles to try to spread them out and equilibrize the concentration on both sides. So that we call that diffusion of water, and we call that osmosis. So water always moves into an area of higher solute concentration. To help you remember that, you can think of solutes suck, and solutes will always suck the water into their location. So this goes through the net movement of water, always travels from an area where there's more water or it's more dilute to a side with less water, less dilute. And water will always move into an area with higher solute concentration. And again, this is showing this little ball that has been dropped into this solution is very concentrated with sucrose or sugar. So water will move into that ball or balloon. And when water moves into that area through osmosis, the balloon will swell. Osmotic pressure is just the force that surrounds the cell required to stop osmosis. It can be used to describe an osmotic pull of a solution. And a higher solute concentration would require a higher osmotic pressure to get the water into it. So here's a look at osmotic pressure. Molarity and molality. A mole of a compound can be measured as its molecular weight in grams, and you can make molar solutions or molal solutions. You probably went through a little bit of this in chemistry. I won't ask you really any questions about molarity and moles in this class because we're not meeting in person, but a molarity is just a way to measure the concentration. Osmolality is the total molarity of a solution when you combine all the molecules within it. And again, I probably won't ask you any osmolality questions. You'll get some of that in chemistry. So I might skip over this part just for now. We will talk about tonicity though. Tonicity is the effect of a solute concentration on the osmosis of water. So I'm going to move into this area. Tonicity takes into account the permeability of the membrane to the solutes. And if the solutes can cross the membrane, the tonicity will change. And I just want to kind of talk about kind of what hypotonic, hypoosmotic, and hyperosmotic mean. So if we look at the fate of red blood cells, if we put them into an isotonic or hypotonic or hypertonic solution, we're just going to talk a little bit of this now. An isotonic solution will be an area around the red blood cell that's an equal concentration to it. So if we put a red blood cell into an isotonic solution, it will retain its shape because it's placed in a concentration of the same concentration. If we put a red blood cell into a hypotonic solution, which is the area around this red blood cell, because through osmosis, water will naturally travel into an area of higher concentration, and the red blood cell has a higher concentration of solute than its hypotonic solution. So water will travel into a hypotonic solution, and the cell will swell and possibly burst, and that's called cytolysis. If we put a red blood cell into a hypertonic solution, a hypertonic solution meaning there's a greater concentration of solute outside the red blood cell, so water will leave the red blood cell and the cell could shrivel up. So you should have a good understanding of these three types of solutions and what a red blood cell will do when placed in either of those solutions. The regulation of blood osmolality, and again, osmolality can be maintained and it should be constant or neurons will be damaged. Osmoreceptors in your hypothalamus, your brain will detect increases in this osmolality due to dehydration. Some triggers would be thirst or decreased excretion in water in your urine. So here's a homeostasis of plasma concentration. If your blood volume from dehydration decreases, your plasma osmolality will increase, and that'll tell your kidneys to try to retain water. It'll also give you a thirst sensation to try to take in more water. So this is a homeostasis of plasma concentration if you were dehydrated. Carrier-mediated transport, molecules that are large or polar cannot diffuse across the membrane, and we need some carriers to help these large polar molecules to get across. Some proteins can transport more than one molecule, but then there's competition, and transport rates will be increased when the molecular concentration and the transport rates, the rats, that's a misprint, will increase with increased molecular concentration. So this is just showing the concentration of something, and the rate of transport will increase if there's a greater concentration of it. Facilitated diffusion is a passive process, and it's powered by the random movement of molecules. It's passive, so no energy, no ATP is used, and there's a net movement from high to low concentration. It will require specific carrier-mediated proteins, and the transport proteins may always exist in the plasma membrane or be inserted when needed. So this is the facilitated diffusion of glucose. Again, it's a passive process. Glucose will go from an area of higher concentration outside the cell to an area of lower concentration inside the cell, but it will use carrier proteins to help it get through the cell membrane, and that's why we call it facilitated diffusion. So this talks about glucose. The transport carriers for glucose are designated GLUT, and they're followed by the number of the isoform, or just the different form of that carrier. This GLUT1 will be found in your central nervous system. GLUT2 is found in your pancreas. GLUT3, this carrier is found in your neurons, and GLUT4 is found in your adipose and skeletal muscle. And again, these are just different transport carriers for glucose, and they're given a different number depending on what type of isoform they are, depending on where they're found. Sometimes molecules must be moved from an area of low concentration to high concentration. Think about they're needing energy because they're moving uphill. So this requires energy, and often these carrier-mediated proteins are called pumps. Primary active transport occurs when the hydrolysis of ATP is directly responsible for the carrier protein function, and the transport protein will also be an ATPase, which is an enzyme that hydrolyzes ATP. And the pump is activated by phosphorylation using a phosphate group from ATP. So what does this mean? Hydrolyzation means the ATP will be broken apart. And when it's broken apart, one of its phosphate groups will activate a pump to help get something through the membrane. The calcium pump is located on all cells in the endoplasmic reticulum of muscles. It will remove calcium from cytoplasm by pumping it into the extracellular fluid, and it will create a strong concentration gradient for rapid movement of calcium back into the cell. It helps to aid in the release of neurotransmitters in neurons, but also in muscle contraction. So this is a look at calcium and just showing how in order to get calcium across the membrane, we need to hydrolyze ATP or break it down. And when we break down adenosine triphosphate, we break it down into an ADP and an extra phosphate group. And that phosphate group energizes this pump to get the calcium across the membrane. The sodium-potassium pump is found in all body cells, and we'll learn a lot about this later on. It has an ATP enzyme that pumps 3 sodium out of the cell and 2 potassium into the cell. I want you to remember that 3 sodium get pumped out of the cell and 2 potassium get pumped into the cell. And sodium is Na, the atomic symbol, and potassium is K. The sodium-potassium pump is important to maintain osmolality. It produces an electrochemical impulse in neurons and muscle cells, and it also provides energy for coupled transport of other molecules. Here are steps of the sodium-potassium pump. I won't ask you guys to know these specific steps, but I want you to know how many sodiums get pumped out of the cell, 3, and how many potassium get pumped into the cell, 2. So here's a look at 3 sodium get pumped out for every 2 potassium that get pumped in. And this is the sodium-potassium pump. And again, it's powered by the hydrolysis of ATP when ATP gets broken up into ADP and a phosphate group. Secondary active transport is also called coupled transport. And the energy needed to move molecules across their concentration gradient is acquired by moving sodium back into the cell. So we call it a coupling transport. Since the sodium was originally pumped out of the cell using ATP, this is considered active transport. Co-transport or symport, the other molecule is moved in the same direction as sodium, and this is a common way to usually transport glucose. Counter-transport is the other molecule is moved in the opposite direction from sodium. So basically, secondary transport can be co-transport or counter-transport. Co-transport means the molecules moved with the sodium in the same direction, and counter-transport means the molecules moved in the opposite direction as sodium. So here's just a look at the extracellular fluid, how sodium and glucose are co-transported into the cytoplasm. And you can see that here. Transport across epithelial membranes happens through absorption or reabsorption. Absorption is the transport of anything digested across your intestines into the blood. And reabsorption is the transport of molecules out of your urinary filtrate back into the blood. So you'll notice when we get to the digestive system, we'll talk about absorption. And when we get to your urinary system, we change it to reabsorption. It involves transcellular transport, which is the movement of molecules through the cytoplasm, and it may also involve paracellular transport. The transport across epithelial membranes involves many types of carrier-mediated proteins, and paracellular transport is limited by junctional complexes. And these are some of the junctional complexes that exist between cells. So this is a look at transport across epithelial membranes, the co-transport, primary active transport, sodium, potassium pump, and then the co-transport, meaning sodium will come in with glucose. And then here is the transport of sodium down its concentration gradient to help get the glucose back in. Junctional complexes, these are tight junctions, desmosomes or macula adherens, and these are junctions between cells that will also affect transport. Bulk transport is when large molecules such as proteins, hormones, and neurotransmitters are secreted via exocytosis, and it will involve the fusion of a vesicle with the plasma membrane, and this always requires ATP. Movement of large molecules such as cholesterol into the cell requires endocytosis, and this is usually a transport protein that interacts with the plasma membrane to trigger endocytosis. So I want you guys to have a good understanding of endo and exocytosis. Endocytosis takes things into the cell. They will fuse with the membrane. The membrane will create its own vesicle, and then the extracellular substances will be now within that vesicle. In exocytosis, the vesicle of usually waste products will join with the plasma membrane, and then it will be spit out into the extracellular environment. Membrane potential, there's always a difference in charge or electricity on each side of the plasma membrane, and that's due to the permeability of the membrane because different things can come in and out of it. It's also due to the action of the sodium-potassium pumps. So these ions have a charge to them, and negatively charged molecules will always be inside the cell. So the inside of cells is normally more negative than the outside of cells, and we call that the potential difference, and that makes the inside of the cell negative compared to the outside. And this is just kind of the basic, really simplified version of the plasma membrane, showing how there's always usually fixed anions, which mean they're not able to be moved. So the inside of the cell always kind of has a negative charge. And what can be moved inside and outside the cell are these positively charged ions. And this is showing the concentration gradient. The positively charged ions will always flow down their concentration gradient, whether that's inside or outside of the cell. But the inside of the cell, because of the fixed anions, is always negative. The membrane potential is due to potassium. Potassium accumulates at high concentrations inside the cell because the sodium-potassium pump actively brings in potassium. The membrane is very permeable to potassium, and the negative anions inside the cell will attract those cations. So the potassium concentration inside the cell is about 150 milliequivalents per liter, and outside is only about 5. So what this just means is that normally there's more potassium, which is K, inside the cell. Even with all these positively charged potassium inside the cell, the negative molecules kind of outweigh that potassium, and the cell is still more negative inside compared to the outside. And this potential difference can be measured as what we call a voltage. Potassium equilibrium, the electrical attraction would pull all the potassium into the cell until it reaches a point where the concentration gradient drawing potassium out matches the pull in. So normal cells just have about 150 of potassium inside the cell and 5 outside the cell. So again, we have an equilibrium of more potassium inside the cell. All I want you guys to know is that there's normally a charge of about negative millivolts inside the cell, and you should just know that number, and that's called the resting membrane potential. It's about negative 90 to negative 70 millivolts. So this just shows the potassium positively charged ions that are being attracted inside the cell due to the fixed anion negative charge, and this gives a voltage of the cell membrane of about negative 90 millivolts, meaning it's charged. The sodium equilibrium, there will be more of a concentration of sodium outside the cell, and that's all I really want you guys to know, is that the concentration of sodium will always be more outside of the cell. So here just shows you the regular or normal concentrations of sodium, potassium chloride ions, and calcium ions, but the big ones are sodium and potassium. And I just want you to know that there's more sodium outside the cell and there's more potassium inside the cell. The Nernst equation, we won't get into this too much, but it kind of goes through some of the equilibrium potentials using a logarithm equation for how to calculate equilibrium potentials. And again, you should just know this shows potassium, which is the K. Inside the cell, it's negative 90 millivolts. Outside the cell with sodium, it's about a positive 66. Even though they're both positively charged ions, remember the inside of the cell is more negative, so that's why the inside of the cell gets more negative. The membrane potential then of the cell not producing any impulses depends on the ratio concentration of each ion. Sodium, potassium, all of these ions with their charges contribute to the resting potential, and the membrane potential of a cell does not produce not producing any impulses because the membrane is most permeable to potassium. A change in permeability for any ion will change the resting potential. This is really key when we get to the nervous system. This will make sense. In most cells, the resting potential is between -65 and -85. So I want you to know that neurons are usually at about -70 millivolts, and that's just the charge at their membrane. When a neuron sends an impulse in the nervous system, it changes the permeability of sodium, meaning that different things will happen where sodium ions will rush into the cell. So -70 is the normal resting membrane potential, and you should know this number. Sodium is more concentrated outside of the cell, and potassium is more concentrated inside the cell. And we'll learn about in the nervous system what will happen as sodium and potassium move into and out of the cell. Again, the role of the sodium-potassium pump acts to counter potassium that are always leaking out. It'll transport 2 potassium in for every 3 sodium out to try to maintain that -70 resting membrane potential. And you should know this. The resting membrane potential, RMP, is -70 millivolts, and the sodium-potassium pump tries to maintain that. Cell signaling. So cells will signal to each other through communication in types, through gap junctions, and paracrine signaling. And I'll let you read through those. They'll signal through each other through synaptic signaling, and that has to do with the neurons in the nervous system, and also endocrine signaling using your hormones in the bloodstream. So these are chemical signaling between cells and just different types, showing paracrine regulators, neurotransmitters, neurons in the nervous system, hormones in your endocrine system between the organs, glands, and bloodstream. Receptor proteins are on a target cell, and that usually receives the signal because it has some sort of receptor protein that's specific to it on the plasma membrane. And this goes through different receptor kind of proteins and what they could signal for. Polar or large signal molecules bind to receptors on the cell surface, and intermediaries are called second messengers. They're sent inside the cell to affect the change. They could be a calcium ion or another molecule. So this just shows how a regulatory molecule, whether it's a neurotransmitter or hormone, what it'll do if a regulator is polar, meaning it's water soluble, it will bind to a receptor on a protein in the membrane, and it'll send a signal to a second messenger, which will take that message to the nucleus. If the regulator is nonpolar, it'll go right through the cell membrane and it'll be able to bind to a receptor within the cell itself. Second messengers, a big one is CAMP, C-A-M-P, cyclic adenosine monophosphate, or cyclic AMP. It's a really common second messenger in the body that you should be familiar with. Steps to activate it: a signaling molecule binds to a receptor, which activates an enzyme that produces CAMP from ATP, and CAMP will activate other enzymes, and the cell activities will change in response. So G proteins, receptor proteins that bind to a signal, and enzyme proteins that produce a second messenger are rarely together. They require something to shuttle between them, which is a G protein. So that's why G proteins are important. They'll be kind of this 3 piece alpha, beta, and gamma subunit, which makes up a G protein. One subunit will dissociate when a signal molecule binds to the receptor and travels to the enzyme. So this is a G protein. It just helps this messaging process. And that's the end of this PowerPoint. Thanks for listening, guys. I know this one was kind of heavy. I tried to skip over what you didn't need to know and focus on what you did. I hope you guys are all staying healthy and well.